Great to have you all here, including several visitors. Are there any introductions? People who aren't usually here, do you want to say who you are? Be introduced? Hello, who are you?
Me?
Yes.
Oh, okay.
You're probably someone who's worked at the NBC for 10 years.
Okay, my name is Kate Meyer. Hi, how's it going? I know. I'm in Julie section. And Ariel is actually my really good family friend from back home in Vermont, so we've known each other for many years, and she came here and I'm here, so.
Welcome, sort of random embarrassment. Sort of breaks the ice. Anyone else I can embarrass? No? I'll stop. Very few seminar announcements, of course, because we're coming up to spring break. There is apparently no IB seminar this week. Wildlife on Friday is Christina Cervantes Yoshida from ESPM. Fish communities in San Francisco Bay Streams and land use. Any other announcements? Anyone? All right. Well, it's my pleasure to introduce Ariel. Ariel is a postbac. She's just finished her undergraduate degree at Stanford in ecology, and spent much of that time starting as a freshman, I gather, working in Liz Hadley's lab, and got sucked into Gopher world. We've got a few gophers here. How many, Jim?
About 21,000.
21,000. You know, there's still gophers out there. You know, so obviously there's a long history of interest in gopher biology, gopherology in the NBC, going right back to our founding days, and then sort of picked up again with Jim and his students. And now, the next generation, Ariel and Liz doing really cool things with them. So Ariel has just graduated. She's currently a, this is a name for it, a biology course associate, like a super GSI, for the human biology course at Stanford, and is thinking about what next. Perhaps a visit to Australia. And that's really tooth or claw, and I'll let her explain the rest. Welcome, Ariel.
Thanks so much. So my name is Ariel Marcy, and I'll be talking mostly today about my work that I did as an undergraduate for my honors thesis. So it's looking at how variations in pocket gopher digging morphology predicts their distribution across California's heterogeneous landscape. So I'll unpack that for you in just a little bit, but I just want to say that a lot of this work I did at Berkeley, so commuting here on the weekends to look at all the specimens that Jim and I guess a few other people got, and also the data. So, what is a Stanford undergrad doing at Berkeley? Clearly, I often wonder this myself. But to explain this, I'm going to start with what's usually at the end of a presentation, with some acknowledgments to some tireless mentors on both sides of the bay. So, first to Liz Hadley, my advisor since freshman year, who spent so much time with me developing this project, and introduced me to her very supportive lab. Scott Fendorf, a soil scientist who literally taught me everything I know about soils, is the second author on a paper that all three of us will be submitting, hopefully soon. And then on this side of the bay, Berkeley, Jim Patton, gopher extraordinaire, whose insights underlied many of the fundamental aspects of my project design. Also, Chris Conroy, the mammal curator who set me up at the NBC, showed me how to do mostly everything, and taught me much else besides. So thank you to these four incredible mentors. I'm here today. But my story starts with a grad student named Jessica Boyce, and she was studying how the small mammal community responded to ancient climate change. So comparing populations of small mammals in the Pleistocene, closer to the last ice age, and then doing modern trapping in the same region to compare today. So two different climates. And so when I was joining her as a field assistant, we were looking at the modern assemblage, so trapping small mammals. So I got to see lots of mice, got experience with measuring, and perhaps one of the most unique skill sets, though perhaps not in this company, is making museum specimen skins. So that was quite exciting. Later in the field season, Liz joined us along with a bunch from the lab. And as a part of this, I also learned about excavation. So I got to do some of it myself. Great. And so all of this was made possible by funding that joins undergrad researchers to graduate students to help them with their dissertations. And I just want to give a plug for any graduate student here. If there's an opportunity to involve an undergrad, please do. It makes such a huge difference, and that's why I'm here. So through this project, I got exposed to all sorts of interesting ideas, got excited about morphology, mammals, distributions, climate. All of this was swirling around in my head when I got introduced to these guys, the pocket gopher. Right, very similar to tuco-tucos. So, like tuco-tucos, the pocket gopher is very versatile subterranean generalists. They build extensive foraging tunnels underground to forage on the seasonally available plant matter. In loamy soils, an adult gopher can dig 10 meters of burrows in a day, which is displacing about 50 liters of soil, which adds up to about 18,000 liters of soil displaced in an entire year. So a lot of hard work. And I just want to note that digging combines both shearing the surface and pushing out the soil material, and so this combined creates a lot of work. And so you see that these tunnels are the smallest diameter possible, which reduces both the surface area they have to shear and the volume of soil they have to dig out. And so because of the very expensive nature of digging, gophers have lots of behavioral and morphological adaptations to reduce the energy required to dig and get their food. And so one of the things that they do is they burrow at about 20 centimeter depth, which is where you have the densest plant matter possible. So this is slow motion, and you all see why this is my favorite YouTube video. Okay, so I focused on the genus Thomomys of gophers, so this is all within the family Geomyidae. Genus Thomomys are the western pocket gophers, the guys that we have. And gophers in general are very diverse. The genus Thomomys, however, is the most diverse group of gophers with respect to both size and digging adaptations. And so this genus breaks down into two subgenera. Confusingly, Thomomys is also a subgenera, so I'll be sure to distinguish when I talk about this. All gophers in this genus are claw diggers, but the subgenus Megascaphes has additional tooth digging adaptations, which allows them to access harder soils. All of these gophers are asocial, so they exclude other individuals from their burrows, and quite competitive. And so we also see this, if we zoom in on that triangle area, at the species level. So these species have very tight boundaries, and they're allopatric. So in one region, you'll have just one type of gopher, with few exceptions. The only exceptions are where you have areas with two very different types of soil. So this gives us a clue as to what might be causing the sort of modern art-like distribution of these gophers. Oh, one other note is that subgenus Thomomys I'll always use to indicate with cool colors, and Megascaphes I'll always indicate with warm colors. I realized halfway through that I was using Berkeley and Stanford colors, so if that helps you remember, then these guys are Berkeley, these guys are Stanford. Okay. And I'm also using the star to note Samuel Cave, so the location of where Jessica's work was, and sort of the center of where my study region is. Great. Okay, so soil seems to be a clue, but there are a few studies on the environmental determinants of these gopher distributions. So that led to my hypothesis, which is that differences in digging morphology will lead to competitive exclusion or competitive dominance of one species over another, given the particular combination of soil characteristics in each of those types of gophers range. And so I'll unpack this over the series of my talk. But basically this breaks down into two trajectories. First is looking at the differences in digging morphology, and I did this with morphometrics, so this is what actually brought me to the MBZ, to look at all those specimens. And then the second trajectory is looking at this combination of soil characteristics, so using GIS to compare where gophers are to the soil attributes where they are located. So I'll, again, talk about those two things, but first, let's talk about morphometrics. So, as I hope the video convinced you, digging is really hard work. And so gophers have lots of adaptations to deal with this. And as I mentioned, there are two different types of digging. First is the claw digging. So again, all Thomomys gophers are using claw digging. And you can see from this diagram that there are lots of lever arms involved, and by measuring the lengths of lever arms, we can figure out the forces that these gophers can exert. There are also important morphological features like crests, and these are where muscles that are doing the work attach. And so these are the things that we can compare between gophers. So here we have subgenus Thomomys, subgenus Megascaphys. A couple things to note is that subgenus Megascaphys are much larger than subgenus Thomomys. And you can see, comparing these two humeri, that there's slight differences in how those crests are oriented. So these are things that we can compare with morphometric studies. Another adaptation is the tooth digging. So teeth are the hardest material in the vertebrate body, and so this is what allows gophers to access harder soils. Important thing about rodents is that they have ever-growing incisors, and so that means that these incisors grow in an arc like this. And so in order to be able to dig, you have to have an angle of percumbency that is not acute. So like this, no good for digging. So angle of percumbency is a measure of a mechanical advantage. So if we just grow this gopher, by expanding the radius of that incisor arc, we've increased the angle of percumbency. Now it looks a little bit more like a backhoe. So this is a morphology that allows gophers to dig with their teeth. And so if we compare these two subgenera again, subgenus Thomomys, exclusive claw diggers can't do tooth digging. They have an angle of percumbency that doesn't allow them to dig with their teeth. However, Megascaphys, just by growing larger, have increased their angle of percumbency and now are able to dig with their teeth. So within, and one other thing I want to mention is, as I said, gophers are reducing the diameter of their tunnel to reduce the amount of soil that they have to shear and the volume of soil they have to push out. And so this growing size in order to use teeth to dig may present a trade-off with this tunnel diameter. So they're larger, they may have to move more soil. Great. Okay. So within the tooth digging gophers, there are other ways to increase this angle of percumbency, and one of these is changing where the root of that incisor exists. And so if we compare two species within subgenus Megascaphys, we have one, Townsendii, that because it's large has a large angle of percumbency. But here, Thomomys' body has this incisor position adaptation, which allows it to have a more percumbent incisor, even though it's smaller than Thomomys' Townsendii. And so within body, we also see variation at the subspecies level. And so I've arranged these 5 subspecies in order of the angle of percumbency. And so I'm just showing this so that you realize just how much variation there is within this species of gophers, within this subspecies of gophers, and within this entire genus of gophers. So, how do we quantify all this variation? Initially, I started off doing linear measurements, so lengths and widths. But one of the things that you lose with linear data is the relationship between those linear measurements. And so this is when I started looking at geometric morphometrics, which is what you use to make a network of points. So here we have a skull and a bunch of landmarks. You can enter those landmarks in a program, and that creates a network of points, and you have a bunch of different species, so you get clusters of points around those landmarks. And then there are several analyses that look at the differences between your specimens. And so this took me to Berkeley to find adult females. So we used female gophers because they stop growing after they have matured, and so you don't get additional confounds with age using females. I aged 500 skulls and then took the pictures of those skulls from 3 different views. Additionally, 100 forelimbs from, again, three different views, so an incredible amount of picture taking. I did it all in the herp room, so I got to see some of the cool Dracos there. And after taking about 2,000 pictures, I was feeling pretty one with the museum. So this is still a work in progress. I still need to landmark those 2,000 photos, but stay tuned. Hopefully we'll see something cool from that. So again, in progress, but there's a lot there. Fortunately, I can still look at the second part of my hypothesis, whether particular combinations of soil characteristics separate these two subgenera of gophers. So again, these subgenera distinctions are a very useful separation because those two subgenera have very different styles of digging. So I can still look at the digging morphology aspect of my hypothesis. Great. What questions are there? Okay. Cool. All right, I'll keep going. So this is to introduce you to my study area. Again, this star represents Samuel Cave, where I began with Jessica. And I chose a study area because the Museum of Vertebrate Zoology has a ton of gopher data in this region. And this allows me to look at gophers that then I can maybe later look at here and quantify their morphology. And so, again, the Museum of Vertebrate Zoology has locality data for each of these specimens. And so if you map those specimens onto the study region, we see that each point is representing a gopher collection site. And so all in all, there are about 3,500 gophers in my study area, which gives me quite a lot of data. Great. I also used a soil database with georeferenced soil attributes. One thing to note is that this isn't continuous data, but each of those attributes are stored in these polygons. And the beauty of GIS is that I can now combine georeferenced data of soil with georeferenced data of gophers, and then I can pull for each one of those gophers soil attributes of its location. And so using this technique, I extracted 7 soil variables. I can rattle them off if you're interested, but basically I just looked at all the soil variables that this database had and looked at the ones that could maybe potentially affect gopher distributions. And then I took those attributes and weighted them over 2 depths. So first the 20 centimeter depth, which is the depth where gophers are spending most of their time digging, so the foraging depth. And then also at a 1 meter depth, so the burrowing level. So in addition to foraging, gophers create burrows at up to 1 meter depth, and they store food at this level, sort of a rodent refrigerator, if you will. And it's also important for them for thermoregulation. So both of these are important measures for the biology of these rodents. And then I also looked at the depth of bedrock. So is there available depth for these soils to inhabit? Great. So I have numbers for each of these 15 variables for every single gopher that the Museum of Vertebrate Zoology has in that region. Okay. And so from this data, there are 3 sets of analyses. First, exploratory analysis, so just picking of those 15, which are the most important, which seem to separate these gophers the best. We can also visualize how these species spread across these various soil attributes. The second is a classification tree, so this is looking at how multiple variables separate these gophers, so looking at more than one at once. And then finally, using powerful chi-square to figure out which of these soil characteristics are most important, so comparing within those important attributes, which are most important. So the first step was making a principal component analysis, and I apologize for the graph it's about. That's what it looks like, but you can see generally Megascaphus is in this oval, Thomomys is in that oval, and by looking at which soil attributes correspond to the principal components, I can figure out which ones are the most important in separating those 2 subgenera. And so what we found is that this principal component is identifying soil attributes that would influence the cost of digging. So the first of all is clay. So as you know, soil is made of 3 component parts, sand, silt, and clay. Clay is the smallest particle of those 3, and it's the hardest to dig in because it's very small and they adhere to one another. So if you ever have a garden, you know that very clayey garden is very hard to work with. And so this is something that the literature focused on as maybe contributing to the distributions of these gophers. And this quote, which you've seen before, has a range from 360 to 3,400, so that's quite a range. That range comes from different types of soils varying in their clay content. So the second attribute was bulk density. Bulk density is a measure of density, and what it's telling you is how compacted that soil is. So the soil will be more compacted, there will be less air between those particles. If it's more dense, it's more compacted. And so clay and bulk density together is what soil scientists consider to contribute to soil hardness. So this is a vague idea of soil hardness. It's a combination of clay and bulk density. A third variable, which is very important to California, is called linear extensibility, which is a very fancy term for shrink swell capacity. And this measure comes from taking a line of soil, so sort of a cylinder, a plug of soil. You wet it, and then measure its length, and then you dry that cylinder of soil, and then measure how much it shrinks. And this is a property given to certain types of clay minerals. So spectite is one that's very abundant in California. And what's actually happening is that there's layers of minerals that expel water and then shrink together. So there's a very particular quality of clay. Right, what questions are there about that? It's important that you understand this. Okay, great. So if you have a high linear extensibility, that means that there's a greater difference between wet soil and dry soil. And when it's dry, it means that it's really hard to work with. An LE above 3% is something that engineers quantify as damaging human infrastructure. So this is heavy stuff. And you can imagine that this would be very difficult for gophers to dig in, as well as difficult for plant roots to penetrate. Again, I want to emphasize that this is a place where climate can influence soil. So this is a latent property in soil that is wet. This shrink swell only happens when it's very dry, which is why you associate these sort of pictures with deserts. Okay, so this is one of the properties that we found very important. And again, it's correlated with clay because a very particular type of clay mineral shows this property. All right. The final attribute we found to be important was, again, this idea of depth to bedrock. And that is important for gophers, again, because they need to be digging burrows at low levels in the soil, because temperatures there are held about constant, about the average temperature for that location. And that is a place where gophers can go because they can't thermoregulate very well, and also they can store food. Great. Okay, so those are the four attributes that we then looked at. And so now we can create maps of gopher locality in relation to clay at 1 meters. And so the light parts of this map show soils that have very low clay, so 20% or less. The medium gray is 20 to 40, and the dark is above 40. So take about a minute, just look at my data, and tell me what you see. Cool. Okay, tell me what you see. Shout it out.
The light gray is less favorable than the darker gray.
Yeah, so light gray is less favorable than darker gray, so we're seeing fewer gophers where it's very dark. What about between the two subgenera?
So Thomomys looks more general.
The Thomomys looks more
Seem to
generalized?
occur across a wider range of clay types.
Yeah, for sure. So, yeah.
I personally don't see a relationship.
Okay. Well, then I hope to convince you with the next 3 slides. So, first of all, now I'm just showing gophers that are found in soils over 40%. There's very few points, so we're seeing that, again, this relationship that gophers tend to not be in soils that are very clayey. These seem to be pretty anomalous. Okay, let's look at the second bin. So from 20 to 40, so it's medium level of clay. I think I see more megascaphous gophers in this bin. So more megascaphous than Thomomys. This area seems to be sort of an anomalous area where we do see Thomomys in this higher soil clay bin. And stay tuned, I'll get back to that. Great. And then finally, here's the least clayey soils. And I think I see more Thomomys gophers in these areas. So I hope I've convinced you. And I want to show one more, just a representation of how gophers spread within soil types. So now we're looking at all 3 parts of soil, sand, silt, and clay. And this polygon is showing the soils available in the study region. And just by looking where these gopher species fall out, we see that they are preferring the loamy soils. They're not accessing like 50% of the soil available in this region, and that they tend to be below this 40% clay mark. Great. So this shows in general, all gophers have a preference for certain types of soils, these loamy soils, but that clay alone isn't totally separating out these two subgenera. So I need to look for other soil attributes that do this. And so it's a combination of these attributes that I've identified that are going to be separating these two subgenera. So that brings me to my second type of analysis, this classification tree, which is recursively separating these subgenera by multiple predictor soil attributes. So I'll unpack that in the next few slides. But this came out in PNS just a couple years ago. A paper was advocating for these types of analyses because they're ideal for environmental variables. So they don't require a normal distribution, and it allows to look at interactions between multiple predictor attributes. Great. Okay, and so this is the tree that I came up with, but let me explain what it means. So first, we start with our entire data set, which has a certain number of megascaphus, a certain number of Thomomys, all distributed in my soil, in my study region. The program then looks for a soil attribute, let's say linear extensibility, and finds a cutoff to split gophers into two different groups. So in this case, we're looking at gophers that are found in soils that have over 1.6% linear extensibility, and gophers that fall into under 1.6. So that looks like this. And so now, the width of those branches correspond to the proportion of total megascaphus that are found in this over 1.6 LE bin. And the same idea here, where this width is the proportion of Thomomys that are found in that area. And so we can see this on the map where points, you see points over here that are not included in points over here. Great. And so now we can continue this, just looking at one of these subsets. Apply a different soil attribute, let's say bulk density. It finds another point to split gophers, so in this case 1.4 grams per centimeter squared, so again it's a density measure. And we're going to split it into two groups again. And so we can see here that pretty much only megascaphus gophers are found in soils that have high LD and a high bulk density, and then we can continue to split gophers that are in this other bin. And so this is what we have so far, and we can continue down the tree. But the trend I want you to take away from this graph is that megascaphus gophers tend to be found in soils that are deeper, have higher clay, higher bulk density, and a higher shrink swell capacity. So they're occupying the more difficult soils to inhabit. If we follow the other side of the tree, we see the same trend with megascaphus tending to be more towards this area with these more difficult soils. And in general, we see the trend that Thomomys gophers tend to be in shallower soils with lower clay, bulk density, shrink swell capacity, so the easier soils. So before we lose sight of the forest by looking at just one tree, fortunately we have a random forest analysis. So the tree that I showed you is just one of many valid trees for this data. And what the random forest analysis does is that it makes multiple trees. So I ran this with 500 or 1,000 trees. And then what it does is it takes out one of those soil attributes and then looks at how the accuracy of that tree, so the ability of that tree to separate the two subgenera, decreases. So if we take out clay, then it decreases in this amount of accuracy. And what this graph is showing is that this that these classification trees can't tell us which soil attribute is most important in separating these gophers. So they're all about the same within that margin for error. So this is when we bring out the big guns, the nested chi-square tests. And so this is the most powerful analyses that I ran on this data. And we're setting this up with a null hypothesis of an even random distribution of gophers with respect to the soil available in the study region. And so what we do for each one of these soil attributes, we figured out what percentage of the study area had less than 3% linear extensibility. And then did that for each one of those soil bins. And then from that percentage of the study area, using how many Thomomys gophers we have, we can generate an expected number, given an even random distribution, to find in each one of these types of soil. And then we can compare this to what we actually see. And given this data, I can run a chi-square just on this soil bin. So this is telling me that Thomomys gophers are found in low LE soils much more, so significantly greater than expected. And we see significantly fewer Thomomys gophers in these higher LE bins than we would expect. I can also do a chi-square for all three bins, all three soil bins, and this number tells me how linear extensibility affects the Thomomys distribution. I can do the same thing for Megascaphus gophers, so again looking within each bin and then looking at how linear extensibility affects that entire genus, or subgenus. And then I can look at both gophers at the same time, so this is giving me an idea of how linear extensibility affects all Thomomys gophers. But the metric that I'm most interested in is how well this soil attribute separates those two different types of gophers. So for that, I zero in on the bins that separate gophers. So it looks like Thomomys is preferring this lower LE, whereas it looks like Megascaphus is preferring this higher LE. So looking at the bins where the two subgenera show opposite trends. And so this gives me a metric that I can compare with other soil attributes. And so I did this for both the 20 centimeter foraging depth and then also the 1 meter burrowing depth. Great. What questions are there? Yeah.
I have one, Ariel, and that is, those numbers are specimen numbers, they're not localities.
Right, so they're specimen numbers.
Okay, what happens, because your soil measurements are based on localities, have nothing whatsoever to do with the sample size from a locality.
Right.
So what happens if you use, do you have enough power with the number of localities to do this same kind of analysis?
I think so. I haven't done it. I can show you at the end of my talk that I looked at whether numbers of gophers was, if there's any correlation between like a high number of gophers in a particular study region, and it seems like there isn't a huge trend between certain study regions having higher gophers than another. Does that answer your question?
No, I mean, why not use the number of localities rather than the number of specimens?
Yeah.
Since the soil data are based on localities.
Yeah, I haven't done that yet.
I guess confidence you'd be worried about non-independence because you've got a whole bunch of samples in one place. They're not truly independent, spatially.
Yeah, again, I can show you the graph at the end that shows that most of these localities just have one gopher. Yeah, great comment. Cool. Okay, so from, oh, just one other thing I want to mention is that these are highly significant, so really, really significant. So I'm confident that the whole locality number of gophers isn't making a huge difference. I think I would see the same trends if I just looked at localities as opposed to number of gophers.
Like numerically? What are you talking about?
So I would see the same relationships between gophers preferring
No, I
certain
mean
soils.
significance.
Level of
Highly significant. What does that mean in a number?
Oh, so p value less than 0.001. That's what the 3 asterisks mean. Other questions? Yeah.
I think part of that issue depends on the scale at which you can measure the soil properties. And that should be somewhere near the scale in which the gophers are there or not there, are responding.
Yeah.
It seems like that you haven't really mentioned the scale at which you can get that mapping these localities with respect to the soil.
Yeah, I forgot to mention in the slide where I had gophers on top of the soil bins. What I did is I created a 2 kilometer buffer, so a 2 kilometer radius buffer around each gopher locality, and then averaged the soil attribute in that area. So hopefully taking into account some error in measuring. What other questions are there?
One more. Do you think people search for gophers at random across soil types?
Probably not. No, but there's been a lot of sampling.
Absence data, I guess, is an issue.
Sure. I mean, with a lot of environmental studies.
I'm going to disagree with that,
Well,
absolutely.
you do it.
You search where you can find gophers. You don't pay any attention to the soil type when you're out trapping gophers.
Great.
You just
Okay, you're
look
looking
for
for where there
mounds,
are
right?
fresh mounds, period. Yeah.
That's fine.
Great.
Yeah.
So we just need to get 100 more gyms, like do a search radius across this entire
Oh, I
solar
think we
region.
should cope
Then
with
I
The cost is too great.
We should be so lucky. Keep going.
Great. Okay, so to summarize all of the findings from these types of analyses, it looks like morphologically distinct groups of gophers, so these two subgenera, are significantly sorting by clay, bulk density, and linear extensibility. And this is true for both depths. So these numbers are for the 20 centimeter foraging depth, and then these are numbers for the 1 meter burrowing depth. So all three of those attributes are significant at both depths. But not depth to bedrock alone. So all gophers prefer deeper soils. But, as I mentioned, or maybe I forgot to mention, is that these three attributes increase with the depth of soil. So water is bringing clay down to deeper depths, so as you go to deeper soils, it's going to have higher bulk density, higher clay content, and with that, a higher linear extensibility because there's higher clay. So that means that the subset of soil available to Thomomys decreases with depth. So the soils at those depths are harder to dig in, which means that Megascaphus is able to utilize those climate-protected depths. So again, the depths where the temperature is roughly the average temperature, average annual temperature for that location. So that means that subgenus Thomomys gophers are more constrained. They need to be digging in these easier soils. And so we see this where it has a significant preference for lower clay soils, and then it's avoiding these higher clay soils. But we see that Megascaphus doesn't have any significant difference between what we would expect. So it seems that Megascaphus isn't as affected by this soil attribute. But when Megascaphus does prefer a certain soil type, it's always coupled with a Thomomys avoidance. So Thomomys has to be absent in order for Megascaphus to prefer a certain soil type. And so this could be because Thomomys gophers are behaviorally dominant towards Megascaphus gophers, so they're excluding Megascaphus where they can access those soils. Or this could be a trade-off between smaller size, smaller tunnel diameter, and Megascaphus having a larger size and a larger tunnel diameter. Either way, this would be another thing to test. This connects back to, again, my original hypothesis that competitive dominance of one species over another will occur because one of these subgenera has the tools to dig in that particular type of soil better than the other type of subgenera. Yeah?
So of course gophers are famous as ecosystem engineers, right? They themselves churn the soil. So if you can imagine a beginning point of sort of stratified soil, loam over clay, pretty common.
Yeah.
Would you expect ecological succession to occur? Because they're modifying the environment as they're occupying it.
Yeah, it's a great question. I'm not sure at the rate at which it changes, but that would be an interesting thing to look at.
I don't know either, but I'm wondering if someone's studied that.
I know it's an active area of research of like looking at gophers as geomorphic agents, so actually changing the soil. Yeah.
Okay.
Cool. Great, so a couple of conclusions I want you to take away is that first, gophers are universally avoiding energetically costly soils, so soils that have incredible amounts of clay, linear extensibility, or bulk density. As the literature suggests, soil is determining gopher range boundaries by soil hardness. My study is more precisely defining soil hardness as percent clay and bulk density. But the major finding of my work is that linear extensibility is very important in separating these two subgenera of gophers. And linear extensibility, again, is the connection between soil properties and the climate of that area. And so this is where we pick up the story with Jessica again. So her study, again, was looking at how ancient climate change had changed small mammal populations. And with another Berkeley grad student, Jenny McGuire and Liz Hadley, they published in Nature about how climate change from the late Pleistocene had decreased small mammal diversity. And again, here's our study region with the Samuel Cave. One of the things that she found was that there was a gopher replacement event in Samuel Cave. So that means that there used to be a subgenus Thomomys gopher occupying this area, and now today it's a subgenus Megascaphus. So what changed from the cold Ice Age environment to the warm modern times? And I'm going to present a synthesis of mine and her work as a hypothesis that over this time period we're seeing a change in aridity. So we're getting more arid from the Ice Age till now. So that means that we're bringing out the latent linear extensibility in these soils. And so here we have a map of the modern distribution of gophers. Again, here's Samuel Cave. And I'll show an animation of one of the quotes from her paper. So let's say in the Pleistocene, the gopher distribution was like this. So Thomomys in blue, Megascaphus in red. So this is what it looked like in the Pleistocene. Again, this is something I made, it's purely speculative, but it's illustrating this quote. Thomomys mazama, this subgenus Thomomys gophers, declined in the late Pleistocene, whereas Thomomys body shows the other trend. And finally today, body has increased in abundance and is now the only gopher present locally in the Samuel Cave. And so if I remove that oval, we see that's the modern distribution of gophers today. And they're present on soils that have a very low linear extensibility. Whereas if we take off this red oval, we see that those body gophers are present in soils that have a higher linear extensibility. So soil that is able to respond to warmer climates by becoming harder. And so I propose that this is a mechanism for the change that she saw in the ancient bone records. And so with that, we're saying that there's evidence of gophers responding, range shifting in response to climate, and that this will continue to happen in the future. So we can model gopher response to future climate change by looking at linear extensibility. And so our prediction is that in areas that have Thomomys gophers but high linear extensibility, we'll see those places taken over by Megascaphus gophers. And I also want to emphasize that soil changes are not included in current climate change models. So if this can affect gophers, then it's also going to affect any other soil organism, including plants. And so this is an important thing to take into account when we're looking at future climate change. And so to end, I just want to leave you with this thought of, this is the map of the current California climate. The blue is representing the Mediterranean summers where it's very arid, so it's bringing out that linear extensibility, whereas the green aspects are the cool climate, the cool continental climate that has a more moister summer, so summers that aren't bringing out that linear extensibility property. If you look at this green area, we see that's where those Thomomys gophers are able to be in those high linear extensibility soils. So if the climate changed to become more arid in those regions, we would predict the Megascaphus gophers to invade the soils currently inhabited by the Thomomys gophers. So with that, I'd like to thank everybody who helped me. Said go for it. Had to slip that in somewhere. And again, just show how much of a collaboration this project was between Stanford and Berkeley. And again, I want to say a huge thanks to the Museum of Vertebrate Zoology for hosting me for almost 4 years, supporting me in this project with their data and invaluable mentors. And thank you for listening to my talk. And for that, I will take any questions, all questions you have.
Yeah.
So could you show a correlation now between current rainfall patterns and the soil properties that are important?
That would be great, and we're working on that with some of the climate data. So looking at both soil and climate.
So, you know, but then many in the audience might not know that there are kind of two fundamental thoughts as to what organizes pocket gophers spatially in terms of different species. And one is competitive exclusion, and the other is what you generally call preemptive occupancy. And you kind of come down on the competitive exclusion side, but it doesn't seem like your data support that. In fact, it really supports occupancy, the occupancy model, in the sense that what is driving the system is increase in linear extensibility, and one gopher's ability to utilize that and the other gopher's ability to avoid it. So it has nothing to do with interactions of the gophers themselves, but it has more to do with the property of the soils that they can occupy or not occupy.
Well, yeah, I guess I'm having trouble understanding why those are mutually exclusive. It seems like if one gopher is very good at occupying the high linear extensible soils, that when the climate change, it's literally pushing the other gopher out.
Well,
so there's a
or
combination.
is it pushing the other gopher out, or is the other gopher being pushed out because it can no longer utilize those soils that are changing?
Oh, okay, yeah.
So that's the nature of preemptive occupancy. If you've got two soil types that both gophers can utilize effectively, whoever gets there first stays there, unless it goes out on some attribute of its own.
Sure.
It doesn't get pushed out by the others. In this case, it's not one gopher pushing out the other gopher actively. It's a passive mechanism, because the soil is creating conditions that one gopher could live in, but the other gopher cannot.
Sure. I think, though, if you look at this map, you do see Thomomys gophers in areas that are far away from those Megascaphus gophers, still occupying soils that have high linear extensibility. So in the absence of Megascaphus in that region, they're able to access those soils, at least in the Megascaphus case.
But that kind of goes against the model that you proposed then. So what is, that temporal series that you showed, what does create the turnover in gophers at San Juan Cave?
Yeah, I guess I'm trying to think of a way to combine them by looking at how the
That's,
soil
that's,
sort of shifts the
I guess
balance
my
between these
argument
two.
is not in favor of one mechanism or the other, but how do you distinguish between those two? Because those
Sure.
are the driving hypotheses behind gopher distribution and have been for decades.
Yeah.
You almost need some experimental
Yeah.
evidence where you manipulate the background, but you also need some behavioral stuff, right? Because bigger gophers probably able to kick ass, right?
Well, there is some laboratory work looking at Thomomys gophers and Megascaphus gophers, and it seems like the Thomomys gophers are actually behaviorally dominant over the Megascaphus, but I'm not sure how much you can take away from the lab study, but that is what the literature suggests.
That's a little surprising, isn't
It is surprising.
How do they do that?
Well, almost every case where the laboratory, didactic encounters favor one species, in the field, it's exactly the reverse. The laboratory is one way, the field is the reverse.
Then that supports your argument that it's environmental rather than.
I know these soil maps have a long history in California, and I've been aware of them for a long time, but I really don't know what the scale is, or how are the maps actually drawn? I mean, they didn't go to every place there. Did they do it with a grid based, or do they just extrapolate from the samples they have, or how is it actually put together?
So they're extrapolating from samples that they take for agricultural purposes. I think they have a pretty accurate record. And so here I can go back to but it's organized in soil polygons. So you can see like this particular soil polygon has the same numbers for each one of those soil attributes as compared to this soil polygon, this soil polygon, and so it's organized in those shapes.
So somebody drew those fine white, they look like fine white lines on a gray background to me, and they drew those in.
Yeah, so I guess a better place to compare is like here's one soil polygon, and here's another, and so they took samples in those areas, and then based on what they know about the geography, then they can extrapolate for that area.
these gophers interact with any other burrowing rodents, like ground squirrels and similar things?
Yeah, I think there's a similar exclusion. There's just one or the other present, just because.
No, I'm asking that because do, for example, ground squirrels will affect one of these two gopher species differently?
I don't think so, because underground, one of the constraints is just how much biomass is available to support a rodent, and they're all going to be digging in the same area to get at.
So if ground squirrels are there, there is no problem for anything.
Right. Yeah. Or it's the same problem for all of them.
Have you looked at vegetation as a consequence of the soil characteristics?
I looked
That
at.
may be a complicating variable.
Yeah, I mean, gophers are generalists, so they tend to eat pretty much anything that they encounter. But would you say that is true?
Relative to other subterranean rodents, yes, they're not geophyte specialists, but they certainly have a narrowed range of plants that occur in the environment that they actually feed on.
Right, but all of these gophers have similar preferences, so it would be.
But there's no difference as far as we know between food habits of any species of Thomomys, except for what's just available to them.
So we're up against 1 o'clock. So let folks head off to whatever they need to do next. I want to thank you for